Good day, this is Professor Will, CBMD PhD. Today is Thursday, October 1st, 2020. It is 6:38 and 30 seconds a.m. Eastern Standard Time, and this is a continuation of my microbiological research. So let's hop to it here. While the closely related agrobacteria and rhizobacteria, which play such important roles in the plant kingdom, are not members of the rickettsial group, the alpha phylum appears antidotally to have been more prone to the horizontal import of genes than are most or all other proteobacteriophyllae, as judged by the number of archaeal genera, amino-acyl tRNA, synthase genes they contain. A systematic study of HGT in the alpha phylum is definitely called for, especially an accounting of when in the phylum's evolutionary history the bulk of these transfers occurred. The photosynthetic phenotype therein, represented in several lineages, is unlike the photosynthesis found in the gamma phylum. The latter are the purple sulfur bacteria, but akin to the purple non-sulfur type seen in the alpha phylum. I am inclined to believe, on the basis of the relative uniqueness of the characteristic beta rRNA sequence, that the grouping may have begun as a rapidly evolving offshoot of the Y lineage. The gamma proteobacteria do live up to the implication of the designation proteo. A great deal of morphological and other phenotypic variation is housed therein. Thus, one can make few generalizations about the phylum as a whole. The gram-positive eubacteria, this ecologically and phylogenetically major eubacterial kingdom. The genus Eubacteria is one of the few classical eubacterial groupings to survive relatively unscathed the molecular phylogenetic assault on bacterial taxonomy. A fair bit of taxonomical reorganization has occurred within the kingdom, and some new members that do not possess typical gram-positive walls have been added. But the two classical traits, endospore formation and gram-positive wall, turn out to be phylogenetically valid characteristics. The only real taxonomic surprise has been the addition of the photosynthetic heliobacteria to the group. The main phylogenetic split among the gram-positive eubacterial separates the high G plus C group, the actinomyces. The phylogenetic split between the two is great enough that their specific relationships might be questioned, while the coherence of the entire kingdom was apparent in phylogenetic analysis of the small subunit rRNAs. It cannot be reliably demonstrated in my experience using the large subunit rRNAs. What is called for here is an appropriate test of the coherence of the kingdom based upon genome analysis of the proper gram-positive genomes key, one of which have yet to be sequenced however. While most eubacterial genomes are sequenced for medical reasons, and this is surely true in the gram-positive kingdom, one would still not single out the kingdom as an evolutionary training ground for pathogens. Metabolic versatility is its hallmark. The Clostridia, not its genus or family, but phylogenetically widely dispersed agglomerate of many different low G plus C gram-positive lineages, are classically notorious for the variety of degradative biochemistries of which they are collectively capable. Several Clostridio genomes have been sequenced, but many more are needed, and I would hope chosen in a systematic phylogenetically comprehensive manner. Good day, this is Professor Will, CBMD PhD on Microbiology, Bioinformatics, and Computer Science. I hope you enjoyed listening. Until next time, have a great day.